We are studying muscles and muscle physiology, and this is part 3 of chapter 9 from our Human Anatomy and Physiology textbook. In the previous section, we learned about excitation of skeletal muscles. How do we activate them, and how does the activation by a motor neuron lead to the actual contraction and shortening of the muscle? Well, now we're going to take a look more at the physiology of muscle contraction, now that we know how it kind of takes place. So we'll look briefly at a disease called Myasthenia Gravis, and we'll look at a couple of mechanisms by which we can control the tension on muscles, right? We know that we can either lift light or heavy objects and so forth. We'll look at a couple of types of contractions, and then we'll discover or investigate where does the energy come from, right? We know that ATP is needed for cross-bridge cycling and contraction, but how do muscle cells generate that ATP specifically? So myasthenia gravis is an autoimmune disease that impacts muscle strength or tension, and it involves attack of the immune system on the acetylcholine receptors in the neuromuscular junction. And as that happens, the acetylcholine receptors are removed from the membrane as the cells attempt to replace them, and the number of receptors declines, leading to muscle weakness. There's just not enough open receptors in the neuromuscular junction to trigger enough action potentials. So it leads to progressive muscle weakness, maybe initially noticed in the eyelids, drooping with time because of the weakness in the muscles. So the receptors are being destroyed and removed from the membrane, and so there's just not enough pathways by which sodium ions can enter the cell to form this depolarization in the neuromuscular junction to trigger an action potential in the perimeter. At least not often enough. Well, one treatment for myasthenia gravis is to use a acetylcholine esterase inhibitor. Acetylcholine esterase inhibitors block the enzyme in the synapse, in the synaptic cleft, that automatically degrades acetylcholine, so that now when acetylcholine is released from the axon terminal of the motor neuron, it kind of accumulates a little bit more in the synaptic cleft, and so what receptors are remaining here on the membrane will be more certain to be bound to the acetylcholine and lead to contraction. This gives a little improvement in contraction. A couple of other examples of acetylcholine esterase inhibitors. Insecticides, some insecticides that are commonly used are acetylcholine esterase inhibitors that when applied to a pest, an insect pest, will paralyze it by causing acetylcholine to flood all of its neuromuscular junctions, and they'll have contractions unabated. Also, some nerve gases, some biowarfare chemicals are acetylcholine esterase inhibitors. There's been some unfortunate examples of terrorist attacks in using sarin as an acetylcholine esterase gas that can paralyze people and kill. Okay, let's now get into a little bit more of the physiology of whole muscle contractions. And we're introducing the idea of a muscle twitch. A muscle twitch represents one stimulation of a muscle sufficient to produce an action potential on the membrane, depolarization of the membrane, calcium release into the cytoplasm, cross-bridge cycling, and then relaxation. One stimulation. So here on the x-axis we see time passing, and on the y-axis we see tension being produced, the force of the contraction. And we see that initially when we stimulate a muscle by releasing acetylcholine into the neuromuscular junction, or if we're in the laboratory we might use an electric shock, there's a delay before we start seeing tension developed. And that latent period results from all the steps in excitation contraction coupling taking place. Right? It takes a while for the wave of depolarization to travel into the T tubules, calcium to come out into the cytoplasm, calcium binding to troponin, troponin moving the tropomyosin, cross-bridges begin to cycle. And something we haven't talked about, there are elastic elements within the muscle that must be stretched until they become taut before we notice much tension being formed in the muscle. So muscle tension will slowly rise as those elastic elements are stretched tight, and then we start to generate some tension between the tendons of a muscle. I'll now look down at this lower graph and we'll discover something interesting about muscles that we will build on in a little bit. and that is that not all muscles are the same. In other words, the muscle cells that make up all muscles are not the same. Look at this muscle twitch produced in the soleus muscle, the primary muscle of your calf that produces plantar flexion. So when you stimulate this muscle, we have the familiar latent period, but then we have tension building up and relaxation. The whole process takes about 200 milliseconds. If we stimulate one of the tiny muscles that guides the position of your eyeballs in your orbits, the entire process of stimulation, tension development, and relaxation takes about 20 milliseconds, about 10 times faster. And then we have a muscle in between, the gastrocnemius muscle, the other muscle of your calf that produces plantar flexion. So the point is, different muscles respond very differently to stimulation, and it's because there are different types of muscle cells that are sort of enriched in these different muscles. Zoomed in version of those slides. Now let's talk about a principle by which muscle tension can be varied. It involves summation, it's called. And this little graphical hump right here is the same thing as one of these muscle twitches. So what we're looking at is stimulation of a muscle, tension being developed, and relaxation, so a single stimulus. And the muscle goes back to rest, zero tension in the muscle cell. If we stimulate it again in exactly the same way, another identical muscle twitch would be produced, and so forth. However, if we stimulate that muscle again, or that muscle cell again, before it completely relaxes while there's still some tension generated there, the same identical stimulus will now result in a greater amount of tension and an incrementally increasing tension. As we stimulate at higher frequencies, we produce greater tension. That process of increasing muscle tension with increasing frequency, number of times per second or per minute that we stimulate, is called wave summation. Low frequency stimulation doesn't produce that effect, but higher frequency stimulation produces wave summation. And in fact, at some point, we reach the maximum tension that a particular muscle or muscle cell can produce. You can do it in either way. Imagine either scenario. So each muscle cell has a finite amount of cross bridges that can cycle, and once we activate all those cross bridges at the same time so that they're cycling and producing tension, there's no way to produce any more tension. The implication, though, is that with a single stimulus and calcium released in the cytoplasm, we do not activate all of the cross bridges. Some of the troponins have calcium bound and allow cross-bridge cycling to take place, and some of them don't. And so if we add more calcium by releasing more from the sarcoplasmic reticulum before it's all pumped back in, we're increasing the concentration of calcium in the cytoplasm, increasing the number of troponins that are activated, increasing the number of actin filaments that are unsheathed by tropomyosin, and increasing the number of cross bridges each time. So in all these cases, the calcium concentration is rising and rising, and tension is rising until we saturate all of the troponins and all the cross bridges are cycling, and that's all the tension that the muscles can produce. And that's exactly how we turn on muscles. When you activate a muscle to contract, you produce a series of stimuli from the motor neuron and sort of finesse the muscle into tension production until it reaches what's called fused tetanus. That's all the tension it can produce. That's how we operate muscle cells. They're either off or they're at maximum tension. So that shows us a way of modifying tension incrementally until we reach fused tetanus, but it doesn't really explain the question of how we can control the tension during a contraction of various types. So for that, to answer that question, we're going to introduce the topic of recruitment. So to understand recruitment, we must define a motor unit. So here's a fictitious muscle in cartoon form, and here's a fascicle being pulled out from that muscle so we can zoom in on it a little bit. And here are all the muscle fibers or muscle cells that make up that fascicle. And we discover that a motor neuron emanating from the spinal cord is attached to a finite group, a specific group of muscle fibers. So whenever this red motor neuron sends impulses to the muscle cells, these 3 muscle cells will be induced to contract, none of the other ones. This purple motor neuron, whenever impulses come down its axon, these 2 muscle cells will contract. So the motor end plates on the cells are permanently affixed to a particular motor neuron, and they contract only when that motor neuron is active. So let's say we develop impulses in this purple motor neuron, a whole series of them, and the 2 muscle cells shown that are attached to it reach fused tetanus. That produces a certain amount of tension. What if that's not a sufficient amount of tension for a task that we wish to complete? Well, then we can add impulses coming down another motor neuron at the same time, and now 3 more muscle cells will begin to contract and reach fused tetanus. Now we have 5 muscle cells doing all their work to generate tension in this muscle, and so on. We can continue that process until we develop enough tension to complete the task at hand. So that's called motor unit recruitment. Each motor unit is defined as 1 motor neuron and the muscle cells that it innervates or activates. One more time, a motor unit is 1 motor neuron and the muscle cells that it innervates. So we have 2 motor units shown here, 2 motor neurons and their requisite muscle cells. And if we continue to add more motor units, we get more tension. Here's another way of illustrating it. This is looking at a cross-section of, say, a fascicle of a fictitious muscle, and the pink circles, the light circles, indicate resting muscle fibers. Dark red suggests contracting muscle fibers. They've reached fused tetanus and are contracting. So if we stimulate 1 motor unit, we'll develop a little bit of tension in that muscle, right? 2 cells are contracting. If we add another motor unit, another motor neuron that's active and all of its muscle cells, now we've got more muscle cells contracting and more tension. And then if we add another motor unit, we'll have more muscle cells added to the mix, recruited to the contraction. We have more tension and so on. Eventually, we reach a point where all of the muscle cells are contracting and there would be no possible way to develop additional tension. That process of incrementing the tension by recruiting more motor units is just called recruitment for short, or motor unit recruitment. That's the primary way that we control muscle tension. Now, I will say that this is not a real-world scenario here. We will never activate all of the muscle cells in a muscle during a voluntary contraction. We don't have the capability of doing that typically. So some of the muscle cells would always be in this light pink form, but we can train ourselves to increase the fraction of muscle cells active during a contraction, but there will always be some that are not doing anything. And we'll maybe trade off. We'll have like a little tag team action happening during a muscle contraction. So recruitment. There's something else about recruitment we should discover, really fascinating. Recruitment doesn't involve adding more and more identical motor units until we get the tension we want. Look what happens. Here's this little green hump represents the amount of tension produced by the first motor unit that we activate in a particular muscle. If there's not enough tension, we of course recruit another motor unit, turn on another motor neuron, but look what happens. The next motor unit produces a greater increase in tension than we would have guessed. That's because each motor unit has more muscle cells in it than the one before. This motor unit that produced this blue tension has more muscle cells, and we started right out talking like that from the beginning. We saw that our first motor unit had 2 muscle cells, second one had 3, just to bring in that idea. So now we have more muscle cells being added, and in addition, the muscle cells aren't all the same. They're getting bigger and stronger as we go. That's not just a sort of random thing that happened with the artist's conception. That's a bigger, stronger muscle cell that's now being recruited as well. And finally, when we get to the biggest motor units, as we continue to recruit muscle cells to gain more tension, we get into very large motor units. We have this almost geometric increase in tension produced so that we don't have to wait around all day recruiting hundreds and hundreds of tiny motor units. So things really start to ramp up in a hurry. And this final motor unit, or the last motor units recruited, involve the largest muscle cells present in the muscle. So the point is, each muscle has a mixture of muscle fibers, and we can recruit them in different stages. So we start out recruiting smaller, slower muscle cells and fewer of them, and we continue to increase the number of muscle cells with each motor unit and increasing the size and speed of those muscle cells. So pretty awesome, huh? We can identify or define a couple of types of contraction while we're talking about physiology of muscle contraction, just to get a little bit more foundation. Muscle contractions, like the one in the picture, in which the muscle is going to lift this load, we call it, 3 kilogram mass, and shorten the muscle, right? As the load is lifted against the force of gravity, the muscle is shortening. That's called a concentric contraction. It falls into a larger category called isotonic contractions. Iso means same, tonic means tension in this application. Same tension. In other words, once we lift that load against the force of gravity, the higher we lift it, it doesn't change in tension on the muscle, but the muscle continues to shorten. So that's a same tension contraction, and it's called concentric because the muscle is getting shorter during the process. Here's a little graphical representation of this. Here's the tension on the y-axis of this upper graph. We stimulate the muscle, and the muscle begins to develop some tension, and the tension increases and increases during this first phase of the contraction until finally the tension is greater than the mass of that load, and we actually lift it. And then no more tension is developed because the load remains a constant 3 kilogram mass. But look what's happening to the length of the muscle. The lower graph shows the length of the muscle, and the muscle is shortening. The length is dropping over time as we lift up that mass. So that's a graphical representation of an isotonic contraction, and specifically a concentric contraction. Well, at least in this region, once we start setting down the load and lengthening the muscle, this phase of the isotonic contraction is called eccentric. E-C-C-E-N-T-R-I-C. Eccentric contractions involve the lengthening of the muscle. We didn't just drop that weight, we're setting it down by a controlled muscle contraction. So that's called an eccentric contraction. How about isometric contractions? Isometric means iso, same, metric meaning length or value registered. I don't know what the word I want is. Anyway, if you have an isometric contraction, there's no change in length. Same length. So in this case, let's say this muscle cannot develop 6 kilograms of tension. It can only develop 5 maybe. And we induce it to contract, attached to the 6 kilogram load. It does in fact contract. Crossbridges are cycling, the elastic elements are being stretched, ATP is being hydrolyzed, but the load will never be lifted because the muscle can't generate that much tension. So we have the similar scenario. Stimulate the muscle, tension rises to a point where now, in this case, it's the maximum tension the muscle can generate. So we have a constant tension. But it's not the same as an isotonic contraction because look what happens to muscle length. Absolutely nothing. You can't shorten that muscle because the tension isn't sufficient to the load. So that's isometric contraction, just so you have a little bit of terminology that you understand about muscle physiology. Okay, so our last topic is the discussion of where does the energy come from? Where does the ATP come from? How do the cells generate that ATP for all these muscle contractions? And shown here in 3 tan boxes are 3 different pathways or processes by which muscle cells can produce ATP. Now, it's not that these things happen in absolute exclusion of one another. They overlap, but just conceptually, it's easier to think of them as separate processes, so that's how we'll kind of tackle it. So when a muscle goes from rest to starting to contract, there's always some ATP already stored in the cytoplasm or ready in the cytoplasm. That's probably a familiar principle to you. Also, though, in muscle cells, there's another high-energy phosphate-containing storage molecule called creatine phosphate. And creatine phosphate can be used to restore ATP in a very rapid way at the beginning of a muscle contraction. So for the first couple of seconds or few seconds of a muscle contraction, as ATPs over here, maybe in some area of the cell, are being hydrolyzed during crossbridge cycling, forming ADP. ADPs can be restored to ATP and then brought back up for more crossbridge cycling by utilizing creatine phosphate and reducing it to creatine. So there's a certain amount of creatine phosphate there that can keep us going for about 15 seconds. If we're going to do a contraction that lasts longer than that, we're going to need more ATP. And so the cell will turn to glycolysis, an energy-producing pathway in the cytoplasm of the cells in which glucose molecules are one of our two main fuels for exercise contraction, is oxidized to produce pyruvic acid and 2 ATPs per glucose. So 10 chemical reactions called glycolysis that result in the production of 2 pyruvic acids per glucose and 2 ATPs per glucose. Now this is called an anaerobic process because it requires no oxygen to do. And that can allow us to continue producing ATP for contraction by very quickly oxidizing many, many glucose molecules for about a minute before we get into a pickle and we can't do it any longer. What pickle is that? If this is the only way we produce ATP for the contraction, then the pyruvic acid is going to have to be fermented to lactic acid in order to keep going for that full minute, or maybe a minute and a half. At the end of that time, we're going to produce so much lactic acid in the cytoplasm of the cell, among other things, that's going to start inhibiting excitation, contraction, coupling, cross-bridge cycling, tension development, and the muscle is going to fatigue. So if all we do is anaerobic glycolysis or respiration, it's a short-lived process that leads to quick muscle fatigue. However, this usually represents the occurrence during a high-intensity contraction, meaning a lot of tension, maximal amounts of tension. What if we do a submaximal contraction, something a little more backed off and gentle, a little less forceful? Well, in that case, we can do glycolysis, break down glucose molecules to form pyruvic acid, but then we can take the pyruvic acid and turn it into acetyl-CoA and introduce it into the mitochondria. And in the mitochondria, run it through a couple of more pathways, namely Krebs cycle and the electron transport chain, and produce 32 to 34 ATPs per glucose molecule. Wow, a big jump in efficiency here. So if we're doing something a little more measured, in other words, low intensity contraction and exercise, we can do aerobic respiration. It does require oxygen when we get the mitochondria involved. But look at the payoff. We get 32 ATPs per glucose, and there's no real limit to how long this can proceed. We can continue to exercise for virtually ever, as long as we kind of train our bodies to do it. It does take a little bit of adaptation of your muscles, but we can just do this all day long, or at least for a much longer period of time. Half an hour, 2 hours, 3 hours. So you get the idea. Short, high-intensity contractions that last for 1 minute or a little more involve anaerobic glycolysis and fermentation and lactic acid production, or lower tension contractions that can be supported by aerobic respiration, getting 32 ATPs per glucose using the mitochondria, using Krebs cycle and electron transport chain. So that's the story, right? If we exercise for a short period of time, we'll use ATP and creatine phosphate for the first 10 or 15 seconds. If we continue to exercise for, say, upwards of a minute, we'll use anaerobic glycolysis typically, especially if we're working at the fastest rate. If we're sprinting at our fastest, we'll be able to do that for a short period of time. You know what's going to happen. We're going to start to fatigue, and very quickly and we'll produce a lot of lactic acid, and we won't be able to run anymore, and we'll take a rest. What will happen during that rest? We'll have to replace all that creatine phosphate and all that ATP and all the oxygen that was in the muscles. Or if we do a submaximal exercise, in other words, jogging along instead of sprinting our fastest, then we can produce ATP by aerobic respiration. We'll utilize glycolysis plus Krebs cycle and electron transport chain in the mitochondria and produce all that extra ATP, 32 ATPs per glucose, and we can just do it indefinitely. That's why you can jog for a long, long time. So that's the story of energy production for contractions in muscles. join me for part 4 as we wind up this chapter talking about the length-tension relationship in muscles, and we'll also move on to talk about smooth muscles. This whole 3-part discussion has been about skeletal muscles, but we do want to introduce smooth muscles and find out how excitation-contraction coupling in smooth muscle cells differs from skeletal muscle cells. So join me for that next time.